I'm Luoxi Zhang. Today I will present our work on bark distance, which are new distance metrics for comparisons of mutation trees. This is a joint work with Katarina Yang and Nico Bierwinkel of ETH Zurich and SIB Swiss Institute of Bioinformatics. Tree structures are commonly used to model the evolution of species, genes, and genomes represent RNA structures. Recently, label trees has been proposed to represent how mutations were acquired in temporary order in cancer evolution. In a mutation tree, the root represents the very first tumor cell, and other nodes represent cell populations involved from the founder tumor cell, which are labeled with novel mutations, first seen in the population. Another type of label trees is annotated gene trees, in which leaves are labeled with gene family members. Internal nodes are annotated with gene duplications and specification event. The right figure represents the duplication history of alpha, beta, gamma hemoglobin genes, where black nodes are duplication event and white circle nodes gene specification event. Our work studied distance metrics for comparison of label trees. We consider three types of label trees. Phylogenetic trees are rooted trees in which only leaves are labeled. First type of tree is a labeled phylogenetic trees, in which every node is labeled. However, leaves and internal node labels are not exchangeable. The second type of trees are one-label trees, in which nodes are uniquely labeled with alignment of a label site. The third type of trees are mutation trees, in which nodes are labeled with disjoint subset of labels. Each label is a gene mutation. Mathematically, one-label trees are a special class of mutation trees. How to quantitatively compare trees is an important issue of tree application in biology. Distance measures proposed for comparisons of phylogenetic tree are not immediately applicable to label trees just mentioned. Recently, novel distance measures has been proposed for mutation trees. However, these measures have their own limitation. No standard approach has yet emerged for measure the difference between the mutation trees. In the rest of this talk, I will first generalize nearest neighbor interchange distance and the Robinson fold distance onto one-label trees. We also present a novel relation between that distance measures. Importantly, we present a bark distance for mutation trees and study by simulation the relations of bark distance with no distance measures proposed recently. Lastly, we present a case study involving 3 leukemia mutation trees before concluding this talk. Now we move to our first result. On a phylogenetic tree, each edge induces a two-part partition of leaf labels. One part consists of the labels of the leaves below the edge. The second part contains the labels of the leaves not below the edge. Since the tree is rooted, the partition is ordered. For example, the right edge induces this two-part partition, 1, 2, 3, verse 4, 5. In fact, all these edge-induced partitions determine uniquely the phylogenetic tree. Therefore, the Robinson fold distance between two phylogenetic trees is defined to be the number of ordered edge-induced partitions in one tree, but not in the other. In 1978, the Bach introduced contraction and decontraction operations on stanna trees, which are arbitrary trees with labeled leaves. Decontraction operation is also called expansion. A contraction merges two single-edge connected nodes into one node. In contrast, expansion operation performs the reverse way. It splits a node with degree at least 2 into a 2 node. The subtrees of the original node are assigned to the two new nodes in an arbitrary manner. It turned out the Robinson foot distance between two trees S and T is equal to the least number of contractions and expansions that are needed to transform S into T for any S and T. Nearest neighbor interchange operation was originally defined on phylogenetic trees. In the case of rooted phylogenetic trees, it allows to interchange two subtrees across an internal edge. For example, when apply the NI operation on the right edge, it allows us to move the subtree L down. They move in the same time, move the subtree T up. Another alternation is interchange subtree T with subtree L. We propose to generalize the NI operation onto one label trace as following. Since a node is of arbitrary degree in one label trace, we allow to interchange an unequal number of subtrees across a branch. For example, on the left, the generalized NI operation interchange one subtree below A with two subtrees below B. Since each node has a label, so we also allow to interchange the labels of the head and the tails of the edge, as shown on the right. Before the NI operation, the head of the right edge is B, the tail is A. After the NI operation, so the head now becomes A, so tail is B. In summary, the generalized NI operation allows to interchange arbitrary number of subtrees across the edge. It also allows to change the labels of the internal edge. With that, we define the generalized NI distance between two trees S and T to be the least number of generalized NI operations that are needed to transfer S into T. Then we prove the following theorem. It says for any S and T with the same number of nodes, the generalized NI distance between S and T is bounded above by the Robinson distance S and T, is bounded below by the half of the Robinson distance between S and T. We now move on to our key result. We propose a bark distance for label trees. Robinson foot distance is popular, a metric for comparison of phylogenetic trees. However, Robinson foot distance is not suitable for mutation trees. Mutation trees can have a different label set. Mutation tree can also have different sizes. For two trees of different label site, the Robinson foot distance between these two trees is simply the sum of edges of either tree. We propose to define the bark distance between two mutation trees by considering the partitions on the site of common labels. We use LS to denote the label site of a label tree S. Then we use C to denote the site of common labels of these two trees S and T. Then we use the PC to denote the set of all two-part partition of S. The PS to denote the set of edge-induced partitions of LS in S. To introduce the bulk distance between the two trees, we need to define the similar relationship first. For edge-induced partition P prime of S and a two-part partition P of the C, the common set of labels, the set of common labels. We see that P prime is similar to P if we restricted on the set C, P prime give the same partition as P. So the bulk distance of ST is defined formally as this. So we use an example to illustrate how to compute the bulk distance between the two trees. Here are two trees, S and T, each has 7 edges. So there are 7 edge-induced partitions of S, which are listed below. There are also 7 edge-induced partitions of T, which are listed on the right. So it's not hard to find out these 3 edge-induced partitions of S and these 2 edge-induced partitions of T are similar to a common partition on the set of common labels, which is A versus B, C, D. It's also easy to see that this partition on S and this partition on T are similar to a same partition on the common label site. That this one and this one also are similar to a common label site. Therefore, by definition, the bulk distance between ST is equal to 7 plus 7, then minus these 3 terms. The first negative term accounting for this factor, then this term accounting to this factor, then this accounting for this factor. So therefore, the bulk distance between these two trees is equal to 10. The bulk distance has the following properties. If the two trees have the same label site, then the bulk distance between them is equal to Robinson-Foot distance between the two trees. If they have a different label site, then the bulk distance between these two trees is bounded from above by the Robinson-Foot distance, S and T. The Robinson-Foot distance is a distance metric. Here we also prove that the bulk distance satisfies the triangle inequality. It's also a distance metric. The bulk distance can also be computed in linear time. Then we further use the bulk distance to define order K bulk distance for label trees, which is motivated by the following fact. One label tree is uniquely determined by its edges, U and V. Therefore, it is uniquely determined by the rooted K-star subgraphs. So we use the K-star subgraphs to define K-bulk distance for label trees. The K-star subgraph below a node consisting of the node, it moves the K distance far from U. For example, in this tree, the one-star subgraph below the node U contains a 3-node 1, 7, 8. So the 2-star subgraph below 3 contains 5 nodes, 3, 1, 4, 7, 8. The order K bulk distance is equal to the minimum of the sum of the bulk distance between a K star subgraph of S and its image K star subgraph of T over all possible one-to-one mappings from the K star subgraph of S to the K star subgraph of T. So this is the formal definition. Through minimal weight matching on bipartite graph, we can also define the order K bulk distance between two trees ST of different sizes. Now we move to our simulation experiment. We have a 3 bulk distance matrix. There are also 5 existing distance measures. We use a simulation to investigate the frequency distributions of pairwise distance for all the distance measures. We also investigate the correlation of these distance measures. We consider all possible pairwise distance values between one label trace of 7 nodes that may have a different root. For bulk distances, there are a small number of possible values. We draw the bar chart graph in figure A. Similarly, we draw the bar chart graph for TD and MLTD in figure C and D. For AD, CASET, and DISC, we bin all the pairwise distance into 40 bins and draw their histograms in figure B. We have 2 observations. The first, the frequency of a bulk distance pairwise distance likely have a Poisson distribution. Second, the frequency of AD, CASET, and DISC pairwise distance have a different distribution from bulk distances. This is also true for TD and MLTD. We examine the correlation of distance measures by using the distance from a 30-node label tree to label trees randomly generated from that tree. We first pick a tree T0, which had 30 nodes. Then generate random trees in 4,000 iteration steps. In each step, we generate 5 new trees by applying the generalized nearest neighbor interchange on a tree from a previous step. In total, we had 20,000 trees. Next, we compute the distance from T0 to all 20,000 trees that may have a different root. This is all the dot plot of distances from the T0 to the other trees for each pair of distance measures. The first 2 graphs, this one and this one, show correlation of bulk distance and 1 bulk distance and 2 bulk distance. The other graphs, except this one, show correlation of bulk distance and the other distance measures. We have 2 findings. The first is AD, DISC, MLTD, and TD correlated well with bulk distance. The Pearson correlation coefficient for them ranges from 0.38 to 0.543. The second, the bulk distance and the CASET were poorly correlated. The Pearson correlation coefficient for them were below 0.112. Lastly, we have a case study. We examined 3 leukemia mutation trees. The pairwise distance of these trees are consistent under different distance measures. For each distance measure, as shown in this table, the distance between tree B and tree C is greater than the distance between tree A and tree B, but smaller than the distance between tree A and tree C. Here are conclusions. we have introduced bulk distance, which can be applied to label trees that are rooted or unrooted. The bulk distance satisfies the triangle inequality are linear time computable. The bulk distance correlated well with AD, DISC, ML, TD, and TD for similar trees, but have a different frequency distribution from the other measures. The bulk distance can be easily generated to the mutation trees that contain recurrence mutations, where labeling subsets are not necessarily destroyed. Another contribution of this work is the connection between the Robinson foot distance and the generalized nearest neighbor interchange distance. This work raises a couple of interesting research problems. For example, do the pairwise bulk distance follow Poisson distribution? Another problem is what is the time complexity of computing the generalized nearest neighbor interchange distance? With that, I stop here. Thank you for attending my talk. I'm happy to answer any questions.